Hello all. My name is Malia Santos, and I want to thank you for tuning into my talk today. Today, I'm going to be presenting you some of my phylogenetic work in Tricholosia and some of our initial systematic and biogeographical implications. So to start, one of the main questions asked in evolutionary biology is understanding what processes have led to the patterns of species diversity that we observe today. These processes, whether abiotic or biotic, all play a role in shaping species diversity. I'm only showing you just a few examples of some variables, but would like to highlight one factor that I am most interested in and what has driven me to work in Rubiaceae, and that is herbivory. So Rubiaceae, being the fourth largest plant family, is mainly a tropical family. The tropics are a known source of biodiversity, where herbivores can act as a source of selective pressure due to the increase in frequency and intensity of the ecological interactions. Rubiaceae also possesses many interesting defensive traits, such as secondary metabolites like caffeine, foliar domatia being structures that plants will make to house certain predaceous or fungivorous arthropods, and foliar bacterial nodules, which is also considered a defensive trait. And even though the tropics provide this really interesting place to study the effects of herbivory on plant species diversity, the tropics, and especially tropical Africa, where my plants reside, is a difficult place to study due to internal and external conflicts, and then also just because of the how much diversity is there. And so here I am showing you a time lapse of botanical collections from 1780 to 2015 by Sosef et al. To illustrate that researchers have collected a majority of places, however, scattered and not continuous over time. You can see this in the yellow to orange points collected from the 30s to about the 70s that cover a broad geographic area. And the red points collected most recently are in concentrated pockets or regions in Africa. And an issue that arises when using older herbarium samples in tropical places is that a lot of the plants were preserved in ethanol, which basically destroys any chances of retrieving viable DNA. So in the phylogenomic era, it's become much more important to have high quality DNA than when we were retrieving just a few plastid loci with PCR. Many studies have relied on these high copy DNA regions like the chloroplast, which poses problems when looking into resolving species trees in complex groups. However, as sequencing efforts have improved, so have our capability to retrieve nuclear DNA from fragmented DNA, which is often the case when you have plant tissue that is not dried in silica gel. And so this capability to retrieve nuclear DNA has resulted in this field of museumnomics, where we can use the herbarium specimen. And it is worth noting here that you are going to end up with some specimen not working as well due to collecting methods used in the past, like ethanol. Sometimes they're just too degraded. Before I jump into our methods, I'm gonna just briefly introduce Tricholosia. So Tricholosia is a genus of tropical woody shrubs or trees that are located throughout sub-Saharan Africa. Most of the taxonomic work done in the group was done by our co-author, Elmar Robrick, who published a series of monographs hypothesizing relationships between species based on morphology and geography. There are 5 sections of Tricholosia, and I won't go into detail now, but I will bring these back up in our results. Except to point out that each of these sections has specific morphological characters that define them, such as sessile anthers in Epidranthera or unisexual flowers in Androgyne. However, this has not been tested in a phylogenetic context yet. Previous phylogenetic work is scarce and limited to a study done in 2009 that investigated the monophyly of Triclacea using 4 plastid regions. They found that Triclacea was paraphyletic and decided to resurrect Empagona to its own genus to solve that issue. Despite the chloroplast data out there, it does not provide resolution at the species level when looking within the group. And so we propose to solve this by adding a nuclear data set to obtain a robust phylogeny. So I'm going to go over my methods very briefly, but I don't want to downplay the amount of time that went into curating this data set. We decided to use the Angiosperm 353 data set that targets or putatively targets 353 single copy nuclear loci. This approach also allows you to recover complete high copy elements that are appropriate for phylogenetic analyses. The Angiosperm 353 has also been shown to generate phylogenetic resolution across various taxonomic levels, which is why we decided to use that. We decided to use Hyde-Piper to process our reads, and we modified our target file using Mickley et al. 2020, along with adding Triclacea samples to improve our capture efficiency. And so before I continue with my methods, I am going to jump to some of my results. This is a taxon by gene matrix that Hyde-Piper provides you, and I'm only showing you this to show you that we had some samples that worked pretty well. And then we had some samples that didn't work well at all. And so because of this, we had to decide what to do with those sequences with low capture efficiency. For this particular presentation, we decided to exclude them completely. And we also decided to exclude paralogs, but realized that there is probably some information in there to be looked at. We've tried various phylogenetic methods, but I'll be showing you the results from my maximum likelihood run in IQ-tree. And so right now I'm showing you our latest tree overlaid with the morphological and biogeographical sections proposed by Robrick. And I want to highlight a few interesting things, such as Empagona coming out as its monophyletic group as sister to Triclacea, which was shown in or it corroborates the 2009 paper on Empagona being its own genus. I would also like to point out that section Problystostemen was proposed as a transitionary section due to some traits that lack the fusion or lack fusion in some of their parts, such as the bracteoles. We can see here that it's probably an early diverging clade and maybe sister to the group, confirming Robrick's hypothesis. We can also see that section Epidranthera is scattered, suggesting that the evolution of a sessile anther probably evolved multiple times. We can also see that section Androgyne forms a monophyletic group, suggesting one introduction into the Malagasy region. This is important because in other groups, they have seen multiple introductions into this region. And so while the morphological sections are not monophyletic and the characters are probably more labile than previously thought, the geography of the species shows some clear distinction. And so the photo on the right is taken from Linder et al. On the evolution of plant diversity and is showing you a broad regionalization of Africa. We can see that the origin of diversity of Triclacea is centered in the Guinean-Congolian region, which was suggested by Robrick. We also see one introduction to the Malagasy region, as pointed out before, and then potentially one introduction here into the Zambesian region or the East South African region. So we have some branches that are not supported as well. And so the next steps for us are to be starting to investigate paralogy using Phylucci. I know Laura Frost has a workflow out there on identifying what she called cryptic paralogs. So we definitely want to look into things like that. We want to filter our data set down more. Of course, we want to increase sampling, and then my broad interests in the group are understanding how plant defenses have evolved through this group. I want to acknowledge all of the botanical gardens that sent me my work. And I would like to just put a little plug in there that Dave Tank just got a job at the University of Wyoming. And so we will be moving out there. So if you're interested in joining the lab, please get in touch with him. Thank you.